right. Good afternoon, class. Once again, we are welcome to our lesson on microbial metabolism. We have been looking in the past lectures, we have been looking at microbial nutrition. But right now, we shall begin our study into microbial metabolism. And we hope that by the end of this lecture, by the end of this lecture series, it's not going to be just one lecture anyway, but we hope that by the end of this lecture series, students will be able to understand the concept of metabolism and what are the various aspects of metabolism, how microorganisms utilize the nutrients for the generation of energy. Now, those are going to be our focus as we go along in this study. So, metabolism. As a word, what is metabolism? Now, metabolism refers to the sum total of every chemical reaction that takes place within the living organism. Now, that living organism in this case is the microorganism. And these chemical reactions that we are talking about, they either result in the release of energy or they will require the intake of energy. But generally, metabolism is the sum total of these chemical reactions. Now, we can categorize metabolism into the different types, the different reactions that constitutes metabolism. Now, the first of those reactions are catabolic reactions, and we are referring to it generally as catabolism. Now, by catabolism, we mean enzyme-regulated chemical reactions, and these reactions release energy. Now, during catabolic reactions, complex organic compounds are usually broken down into simpler ones. So catabolism is actually a breaking down process. In this breaking down process, the complex organic compounds are broken down to the smaller constituents of that organic compound. So by energy terms, we'll say that catabolism catabolic reactions are exergonic in nature. By exergonic, we mean that they produce more energy than they consume. Now, these catabolic reactions, examples include how cells break down sugars. When a particular cell takes in the nutrient, the sugar, this sugar eventually is broken down to release energy. Associated with the energy is CO2 that is given off and water that is released. So that is to say that the organic compound sugar is broken down. And at the end of the day, these simpler substances, CO2, H2O, is released in addition to energy. And so because this is a breaking down process, we refer to it as catabolic reactions. Now, the second type of metabolic reaction that we can come across is anabolism, or we can also say anabolic reactions. Now, any reaction that is enzyme regulated requiring energy are anabolic in nature. Now, when complex organic molecules are built from their constituent, this will constitute anabolism. So any building up process is anabolism. And because it is a building up process, they are endergonic in nature. Endergonic because they consume energy. Now, energy is needed to build up. So these anabolic reactions are endergonic in nature. An example include the formation of protein from amino acids. The amino acids are the building blocks, and when they are built up to form proteins, it constitutes an anabolic reaction. Nucleic acids also can be built up from the constituent nucleotides. So we can call these nucleotides the monomers that come together to build up the nucleic acid. It is also a form of anabolic reaction. And also we have simple sugars building up polysaccharides. Now, all these are examples of building up processes, and they constitute anabolic reactions. So catabolic as well as anabolic reactions is what is summed up as metabolic processes that can take place within the microorganism. Now, ATP, the concept of ATP becomes very vital at this point. Now, in nature, every of these systems, I call them biological systems, in every of these systems that metabolic reactions take place, remember that the metabolic reactions are a combination of anabolic and catabolic reactions. Now, nature does not accommodate a vacuum. Now, what happens is that in a living system where metabolism is taking place, energy that is required to drive the anabolic reactions are derived from the catabolic reactions. So the catabolic reactions generate the energy that drives the anabolic reactions. Now, where does ATP come in here? Now, ATP stores energy that is derived from the catabolic reactions. So when the catabolic reactions take place, energy that is released is stored in the form of ATP, and it is released to drive anabolic reactions. And this eventually, brings about performance of other cellular work. So we can say that the ATP is acting as an intermediate molecule. ATP is an intermediate molecule that acts as an intermediary between anabolic and catabolic reactions. So if I am to, make a little bit of explanation further, the energy that is released during catabolic reactions are not lost. They do not get lost into space. This energy is channeled into driving anabolic reactions that requires energy. And so in that sense, we would say that the anabolic and catabolic reactions in every system are coupled. They are coupled to each other. And ATP is acting as the coupling agent or the intermediary, the intermediate compound. Now, what we have here is the structure, not really the structure, but a representation of the ATP. Now, ATP, as we know, is adenosine triphosphate. And so these are the constituents. This is the ribose sugar, and this is adenosine. And we have the three phosphate groups that constitutes or makes up the triphosphate. Now, if this bond here is broken, it means that what we would have is adenosine diphosphate plus P. Now, and when this bond is broken, a lot of energy is generated. Now, it is this energy that has been stored that will be used in driving anabolic reactions that we're talking about. If, on the other hand, the bond is broken at this position, energy is also released, but not as much as what will be released if this terminal bond here is broken. So ATP, as we said, is acting as the intermediate molecule that picks up the energy that has been released during catabolic reactions and used to drive anabolic reactions. Now, whenever chemical reactions occur, whether it is anabolic or catabolic in nature, enzymes facilitate these processes. Now, what are enzymes? By definition, enzymes are substances that can speed up biochemical reactions. And as they speed up these biochemical reactions, they themselves do not get permanently altered. Okay? Now, enzymes is a term that is used in the biological sciences. If it were to be in chemical sciences, we would have referred to it as a catalyst. So enzymes are actually organic catalysts. In living cells, these enzymes serve as biological catalysts. Now, something else about the enzymes is that they are specific in nature. So a particular enzyme acts on a particular substrate. Enzymes are specific and acts on specific substances called enzyme substrate. There are different levels of specificity. We are not directly studying enzymology right now, but there is this level of specificity. So it follows the lock and key principle, because a particular enzyme will recognize its substrate that it acts on. Now, that is how enzymes are. Under optimum condition, enzymes can catalyze reactions at rates that ranges from 10 raised by 8 to 10 raised by 10 times higher than if the reaction was to take place without the enzyme. So that is to show how efficient these enzymes are. If they could speed up reactions to the tune of 8 raised by 10 times more than if the reaction was to take place without the enzyme. Now, one term that is used in describing an enzyme is what we refer to as the turnover number of the enzyme. By the turnover number, we mean the maximum number of substrate molecules that a particular enzyme molecule can act on and convert to product each second. So in 1 second, how many substrates can be acted upon by an enzyme and converted to product? That is the statement of the turnover number for a particular enzyme. The turnover number usually is between 1 and 10,000, but sometimes they could be as high as 500,000. If I use this 500,000 in an illustration of what the turnover number is, it means that this enzyme that has a turnover number of 500,000 can actually act upon 500,000 substrates, converting them to their equivalent products in a particular second. That, of course, talks about a very, very effective enzyme there. Now, what makes up an enzyme? Okay. What makes up an enzyme? What are the components of an enzyme? Now, some enzyme consists entirely of proteins. Enzymes are proteins. Okay, if we are to use that term, enzymes are proteins. But sometimes we can have just the proteins. But most of the time, there is the protein constituent of the enzyme in addition to another component. So most of these enzymes consist of both a protein portion. That protein portion, we refer to it as the apoenzyme. The apoenzyme is the protein component of the enzyme, while the non-protein component of the enzyme is referred to as the cofactor. So that is to say that the complete enzyme consists of the apoenzyme and the cofactor. Ions of iron, zinc, magnesium, calcium, these are all examples. Remember when we're talking about the nutrients, remember telling us that these substances can act as cofactors. So these are examples of cofactors. If this cofactor is an organic molecule, then we refer to the cofactor as a coenzyme. So the coenzyme or cofactor, as the case may be, is usually attached or conjugated with the apoenzyme to form the full enzyme. So together, the apoenzyme and the cofactor forms the holoenzyme. And by the word holoenzyme, we refer to the entire enzyme. If I want to put this mathematically, so to speak, we'll say that the apoenzyme plus the cofactor. Remember that if this cofactor is an organic molecule, we can refer to it as a coenzyme. So the apoenzyme and the cofactor is equal to the holoenzyme or the complete enzyme. Now, many coenzymes are derived from vitamins. Two of the most important coenzymes that we find in cellular metabolism are nicotinamide adenine dinucleotide as well as nicotinamide adenine dinucleotide phosphate. In short, we usually refer to them as NAD and NADP. Now, these contain derivatives of the vitamin B, which is also called nicotinic acid or niacin. NAD NAD plus, is involved in catabolic reactions. So when we have catabolic reactions, energy yielding reactions, NAD mediates. And remember that these catabolic reactions produces energy. The energy can now be picked up. And in this case, NAD can act as what will pick up this energy. Now, NADP is involved in anabolic energy requiring reaction. So if a reaction needs the energy that has been released by catabolic reaction, it is the NADP that supplies that energy. So why NAD is involved in catabolic reactions, NADP is involved in anabolic reactions. Now let's talk a little bit about how energy is produced itself. In subsequent classes, we will look more closely at them. Right now, we'll take a general overview of the energy producing mechanisms. Now, there are two general aspects of energy production. These two aspects are, one, the concept of oxidation reduction, and then the mechanism of ATP generation. So in one hand, we have the concept of oxidation reduction reactions, and on the other hand, the mechanism of ATP generation. We'll look at them one after the other. In oxidation reduction reactions, as the name implies, they are reactions that involves oxidation coupled with reduction. But what is oxidation? Our rudimentary chemistry makes us know that oxidation is the removal of electron from an atom or molecule. Once electron is removed from an atom or from a molecule, that molecule has been oxidized. On the other hand, reduction is the addition of electron to an atom or to a molecule. Now, why we are calling it oxidation reduction reaction is because when we have a molecule that electron is removed from and that molecule becomes oxidized, that electron is transferred to another molecule which becomes reduced. And that process of reducing that other molecule by the addition of electron is called reduction. And they occur together. And it's the same language I used initially for catabolic anabolic reaction that I said they are coupled in nature. That is how oxidation reduction reactions are coupled. Because what is released from one is used to feed the other. Now, the second concept is the generation of ATP itself. Much of the energy that is released during oxidation reduction reaction is trapped within the cell by the formation of ATP. How does this happen? A phosphate group is added to ADP with the input of energy, and this results in the formation of ATP. This process by which phosphate group is added is usually referred to as phosphorylation. So phosphorylation by definition is the addition of a phosphate group to a chemical compound. Organisms use 3 different mechanisms for phosphorylation. In other words, there are 3 mechanisms that is involved in the coupling of inorganic phosphate with ADP to form ATP. 3 mechanisms are involved. Now, what are these 3 mechanisms? Number 1 is substrate level phosphorylation. Now, substrate level phosphorylation is a phosphorylation that is coupled with an hexagonic breakdown of an organic substrate molecule. Now, I defined it here as when ATP is generated when a high energy phosphate is directly transferred from a phosphorylated compound. That compound is a substrate, and from that compound is directly transferred to ADP. So when you have a phosphorylation that is coupled, linked with the hexagonic breakdown of an organic substrate molecule, we have substrate level phosphorylation. So for every substrate level phosphorylation occurring, there is a high energy organic substrate molecule. And when this molecule is broken down, energy is released. And what happens is that that energy is used for the coupling of inorganic phosphate to ADP for the formation of ATP. Now, the second form of phosphorylation is oxidative phosphorylation. Now, in oxidative phosphorylation, electrons are transferred from organic compounds to one group of electron carriers. Usually, these electron carriers are NAD and FAD as well as their derivatives. Oxidation or oxidative phosphorylation usually takes place in the mitochondria of the cell. And if it is a prokaryote, I mean, like in the bacterium that does not have the mitochondria, it takes place in the cell membrane. And there is usually the electron transport system. Now, when energy needed for phosphorylation is obtained by movement of electrons in the electron transport chain, this is what brings about oxidative phosphorylation. So, once there is an electron transport chain, electron being transferred from one carrier to the next, as this process of transfer continues, what happens is that energy is released in quantum. And when a given amount of energy has been released that is sufficient for phosphorylation, it is channeled into the phosphorylation of ADP. So this is oxidative phosphorylation. And the last is photophosphorylation. And as the name implies, it occurs only in photosynthetic cells. These cells contain chlorophyll that helps in trapping light energy. Just like what happens in oxidative phosphorylation, there is an electron transport chain that is involved here. But we must note that what traps the energy initially is light. The chlorophyll that traps in the form of light. So it occurs, photophosphorylation occurs in photosynthetic cells. So these are the three major mechanisms by which ADP becomes phosphorylated for the formation of ATP. In microorganisms, various means of metabolism exists. Now, one of which is anaerobic respiration. And in anaerobic respiration, microbes are capable of using all sorts of other terminal electron acceptors apart from oxygen. Now, I mentioned the electron transport system just now. Now, what happens in the electron transport system, as I said, is that electrons are transferred from one electron carrier to the next. Usually in aerobic respiration, the last electron acceptor, which we call the terminal electron acceptor, is oxygen. That is what makes it aerobic respiration. So in aerobic respiration, the terminal electron acceptor is oxygen. But in the case of anaerobic respiration, the terminal electron acceptor are some other molecules apart from oxygen. Okay? Now, there is also fermentation. And fermentation, when we hear the word fermentation, what comes to our mind most of the time is, production of alcohol, the industrial definition of fermentation. Well, but generally, fermentation is a type of heterotrophic metabolism that uses organic carbon instead of oxygen as a terminal electron acceptor. So organic carbon is what is acting as the terminal electron acceptor instead of oxygen. Many organisms can use fermentation under anaerobic condition. What I must say here is that microbial fermentation is not a very efficient mechanism for energy generation. More energy is generated during aerobic respiration. But if the microorganism finds itself under anaerobic conditions, well, the organism has to survive. So this becomes, an adaptive mechanism, a survival strategy. So in as much as less amount of ATP will eventually be generated, but at least the organism can maintain its existence. It can remain alive. So many organisms can use fermentation under anaerobic conditions as well as aerobic respiration when oxygen becomes present, because it is more effective. When oxygen is present, more energy will be released for the microorganism. So when oxygen is present, it will undergo aerobic respiration. But when oxygen is not available, then it will opt for anaerobic means of respiration. Now, when you have an organism like this that can swap between aerobic and anaerobic processes, we refer to them as facultative anaerobes. It is unlike the obligate anaerobes that will not survive in the presence of oxygen. Now, fermentative organisms are very important industrially because they are used to make many types of food substances. The different metabolic end products that are produced during fermentation, that is brought about by specific bacteria, is what is responsible for the different tastes and properties of each food. So when you have a particular food, you've heard about fermented locust bean, fermented ogi, fermented, different. What is the end product of that fermentation exercise is what will inform the taste of the fermented food substance. Now, the two main types of fermentation, let me quickly say this here, that this alcoholic fermentation and lactic acid fermentation, we're referring to it as the two main types. However, we should know that there are other forms of fermentation. We have butyric acid fermentation, propanoic acid fermentation, but the two main types of fermentation are alcohol fermentation and lactic acid fermentation. The name we give the fermentation is just the name of the end product of that fermentation exercise. So if the end product of the fermentation exercise is alcohol, then we will refer to that fermentation exercise as alcoholic fermentation. And if the end product of the fermentation is lactic acid, we refer to the fermentation exercise as lactic acid fermentation. Now, this is a representation of the two main types of fermentation. Now, what we have here is glycolysis, where the sugar is broken down initially to form pyruvate. Pyruvate or pyruvic acid is the end product of glycolysis. Now, but this pyruvate can exhibit lactic acid fermentation, or it can exhibit alcohol fermentation. The end product here is ethanol, which is an alcohol, and the end product here is lactic acid, which is lactate or lactic acid. So if it is this pathway that is followed, then it is lactic acid fermentation. But if it is this pathway that is followed, it is ethanol or alcohol fermentation. Now, either of these, both of them, usually will take place when the oxygen tension is low. Lactic acid fermentation occurs more in animal systems. Like, for example, in the case of you and I, for instance, if we engage in strenuous exercise that depletes our oxygen tension such that there is anaerobiosis setting up, this is what happens. Lactic acid will be produced. This can accumulate in muscles, and that is what results in muscle cramps during strenuous exercises, is the production of lactic acid. But in plants, on plant-like organisms, this is what happens, that same condition of reduced oxygen. Ethanol will be produced, which is an alcohol. Now, both types have the same reactant. What are the reactants? Pyruvic acid and reduced NAD. And both are products of glycolysis. Just as I said, glycolysis breaks down this process. We'll consider it in our next lesson. So glycolysis breaks down the sugar, and the end product is pyruvate. In alcoholic fermentation, the major product is alcohol. Carbon dioxide is given off. While in lactic acid fermentation, the major product is lactic acid. What we are going to discuss in subsequent class is for us to see these various processes, and that will be in our next lesson. Thank you very much for your time. I'll see you in the next class.